Gestation is an aggressive, high-stakes process of system integration that begins the moment a blastocyst embeds itself into the uterine wall. This creates an immediate biological engineering problem. The mother and the fetus are two distinct biological systems that must now interface and synchronize. They are required to share resources and exchange gases constantly, yet they must do so without triggering an immunological rejection of the fetus by the maternal host. The biological solution is the construction of a temporary, highly specialized organ, the placenta. The placenta functions as both a physical structural bridge between the two bodies and a chemical control center for the pregnancy. Understanding gestation requires viewing the placenta as the active command center responsible for executing the entire embryonic build. The system build begins immediately after the blastocyst successfully implants. The trophoblast, its outer layer, begins a rapid, aggressive expansion into the surrounding environment. The trophoblast generates finger-like projections called chorionic villi, extending into maternal tissue. These fetal villi and maternal tissue interlock, creating a seamless connection. This mesh forms the placenta, establishing a direct line to the embryo through the umbilical cord. The placenta manages the primary supply chain, facilitating the inward flow of maternal oxygen and nutrients. Simultaneously, it handles waste management, removing embryonic carbon dioxide and excretory materials from the system. This physical interdigitation networks the two systems' supply lines, while keeping their distinct bloodlines from ever directly mixing. Beyond its structural role, the placenta functions as a massive, active endocrine gland. It introduces unique chemical signals into the maternal blood, specifically human chorionic gonadotropin, or HCG, and human placental lactogen, known as HPL. In the later phases of the sequence, the ovary also secretes a unique hormone called relaxin. These three hormones, HCG, HPL, and relaxin, are produced in the female body only during pregnancy. The placenta also forces a multi-fold increase in existing maternal hormones, including estrogens, progestogens, and cortisol. This massive hormonal surge is required to alter the mother's metabolism and maintain the structural integrity of the pregnancy. These hormones act as chemical software, overriding the maternal host's normal functions to force biological prioritization of the fetal system. While the placenta manages the environment, the assembly of the organism occurs within the inner cell mass of the embryo. This mass contains potent stem cells with the capacity to generate any tissue or organ in the human body. These cells stratify into three primary germ layers: an outer ectoderm, an inner endoderm, and a middle mesoderm. These three layers represent the complete physical blueprint for the developing adult. The execution of this blueprint follows a strict 9-month timeline. After 1 month of pregnancy, the first major milestone is reached, the formation of the embryonic heart. By the end of the 2nd month, the fetus develops its limbs and digits. At 12 weeks, the end of the first trimester, most major organ systems and the external genitalia are established. During the 5th month, the system adds sensory and aesthetic refinements, including the first fetal movements and head hair. By week 24, the eyelids separate, eyelashes form, and fine hair covers the body. By the end of 9 months, the blueprint is fully executed. The biological objective now shifts from growth to expulsion. Parturition is the mechanical delivery of the fetus through vigorous uterine contractions. The induction signals for this process originate from the fully developed fetus and the placenta, triggering a specific neuroendocrine mechanism. This starts the fetal ejection reflex, where the fetus signals the uterus to begin mild preliminary contractions. These initial contractions trigger a signal that travels to the maternal pituitary gland. The pituitary responds by releasing oxytocin into the system. Oxytocin acts on the uterine muscle to create stronger contractions. This creates a positive feedback loop. Stronger contractions stimulate more oxytocin, which in turn drives even stronger contractions. This powerful reflex builds until the mechanical force is sufficient to expel the baby completely through the birth canal. Parturition functions as a system termination protocol. Once the baby is delivered, the maternal body expels the temporary placenta, severing the physical link between the two systems. Although the physical bridge is gone, the maternal system continues to support the newborn. During pregnancy, the mammary glands underwent differentiation to prepare for postnatal support. Towards the end of the term, these glands begin the process of lactation. The milk produced during the initial days of lactation is called colostrum, containing a specific payload of antibodies essential for developing resistance in the newborn. Even after physical separation, the maternal system continues to execute a chemical software transfer, ensuring the new biological system is equipped to survive independently.